and welcome. Today, I would like to invite you to tour a laboratory established in Piotrków Trybunalski thanks to the cooperation of three entities. Not only was the cooperation of financial nature, but also, most importantly, both conceptual and mental. The laboratory, which you will see in a moment, is strictly a research and development facility, and I would like to show you briefly its amazing potential. Please step inside. Both entrance and exit from the facility are closely monitored, of course. As you can see, I used this opportunity to change my outfit to a more appropriate one. And now, I think we could discuss a few details and a few techniques that are used in our facility. Let's start with survival, morphological and physiological analysis of research carried out on plants. Thanks to the state-of-art equipment, we can visualize and digitalize the morphological image of plants. This morphological image can then be analyzed by means of various types of software, in order to determine the severity of chlorosis or necrosis that appears on plants. We are able to study various aspects related to plant yield and efficiency, based on the chlorophyll content. As a result, we know how the plants behave in appropriate tested conditions or after exposure to certain substances. We can analyze all of this. Of course, we also have other spectrometric equipment that allows us to optimize and make use of the best conditions to accelerate the growth itself, the ontogenesis of plants. Of course, such physiological research is only the basis for a more detailed study of plants. For that purpose, various microbiological techniques and molecular biology techniques will be used. Microbiological techniques are part of the preparation of specialized planting media, require conditions that ensure sterile working conditions. These tests are conducted in laminar chambers where such conditions are maintained thanks to a filter using an UV lighting. All these experiments, the bacterial and mushroom cultures we can grow, they can be used in various ways. Not only can we use them in the context of identification of microorganisms, but also, for example, the impact of microorganisms on the growth and development of plants, or the emergence of disease that often plague our crops. Another part of the research is the experiments in which we use nucleic acid, and based on the analysis of the presence of nucleic acids, gene expression or the presence of RNA itself, we can determine the response of plants to various factors, be it physical or chemical. Starting from scratch, we can isolate nucleic acids, analyze them, validate them, and then very precisely and very accurately determine, for example, the expression of genes that are active at a given moment at a given stage of plant's life. Of course, this requires specialized equipment. As you can see, the chambers for the preparation of the reaction mixtures and PCR assembly are also highly specialized. As a result, proper sterilization level is maintained all the time, and the chambers do not allow our genetic material to be contaminated in any way. of this type of research requires high-quality facilities. We need certain conditions in order to prepare sterile materials to make the planting media, grow microorganisms in special incubations and shakers. We also need a facility where various types of refrigerators may be used to store the material. Biological material can be rather unstable. In order to preserve this material, we must use either liquid nitrogen or low-temperature freezers. Of course, what would plant research be without the possibility to actually grow the plants even in artificial conditions? We use special rooms for growing plants, phytotrons, phytotron chambers, where we can completely control the conditions, not only temperature and humidity, but most importantly, the lighting. The choice of a lighting gives us a variety of growing conditions, but also enables us to compare different chambers and see how light and other factors affects plant growth. As you can see, each chamber is equipped with a camera system. These cameras collect the visual data continuously and allows us to present the growth rate and the growth of plants themselves during the experiment frame by frame. We have 4 such chambers, one of which will be prepared only for production conditions, while the other 3 can be used to conduct strictly research-oriented experiments. As you've seen in this short presentation, our laboratory can carry out various types of analysis, from psychological and microbiological to advanced molecular biology techniques based on the study of nucleic acids. I hope that you've found our presentation interesting, and we're looking forward to cooperating with you.